The Seralogix Study Manager is the best way to design, execute, and report on your preclinical studies. Preclinical animal studies are very complicated and highly susceptible to errors. World-class researchers who excel in scientific methods regularly get tripped up by the complexity and nuances of statistical models and experimental designs, and by the details of study execution, especially when delegating the details of treatment administration and sampling to a team. This results in animal study findings that are not reproducible in clinical trials or publication of misleading results. And this means a lot of wasted money, and perhaps more importantly, an enormous amount of wasted time the researcher can never recover. Seralogix helps researchers at each step of the preclinical study process to avoid the errors and the small mistakes that can render study results invalid, so that the researcher's ideas and hypotheses can be fully realized with significantly higher rates of reproducibility and ultimately greater positive impact on the problem to be solved. To understand how it works, let's start by looking at how Seralogix supports study experimental design. The study design module presents a comprehensive and structured set of steps to help you think through and capture all the key information and design decisions for your study. It starts with the description step, in which you document basic information, such as the type of study, the hypothesis to be tested, purpose, and intervention model, collaborators, and proposed dates for the study. Moving on to the condition step, we define comparative conditions and treatments to be employed in the study. The researcher may set up one or more treatments or control groups. Here we title the group as Drug X, treated, and assign it as a treatment group. The sex of the subjects in the group are selected now as both, to indicate both male and female subjects will be used in this group. Likewise, we add another group, labeled as untreated, and define it as a control type, then as a negative control. Here again, both is selected for the sex of the subjects as is in the treatment group. In some cases, the same subjects will be used in both the control and treatment groups. If so, we would select the box for treatment and control groups share same subjects. Next, we define the treatments or interventions that will be employed in the study. For example, we name this treatment Drug X and select the method of administration and the dosage that is planned. Next, the researcher may specify information about the animal subjects for the study, including the species, as well as detailed strain information. Strains may be selected from databases sourced from the major labs, or the researcher may specify custom strains for non-standard subjects. And other important information may be documented, such as required age and weight ranges for the animal subjects. Information about the environmental factors for the study are documented next, including things like housing and enclosures, ventilation, temperature, illumination, diet, and even restraint methods. Information about the facilities captured, including any assignments of groups to different rooms in the facility. The source of the animal subjects can also be documented. Here we select an outsourced vendor. Common vendor information is pre-populated to choose from. The next step, the researcher specifies a number of important factors fundamental to the study design, such as possible secondary and blocking factors and the primary outcome response factors. Next, any secondary factors the researcher wants to test for possible interactions with the primary treatment are entered. Here we title it stress. Both continuous and categorical factors are supported. For a secondary factor like stress, the researcher might create levels such as low and high stress. Blocking factors that may introduce variability in the study results may also be tracked and controlled for, and like secondary factors, may be defined as either continuous or categorical factor. Here we define subject sex, male and female, as a blocking factor. And finally, the researcher can create one or more outcome response factors to measure the impact of the treatment being tested in the study. The two groups we created earlier are both selected here, since the response will be measured on each group. Outcome response factors can be selected from major lab sources, or the researcher can define custom response factors as needed. Let's make a custom outcome response. Here, the researcher can choose continuous or categorical as a type of measurement, then defines a method and description, such as pupil dilation. Next, the cost of the outcome measurement is entered. We then need to enter the population means and standard deviations, as well as the best estimate the researcher expects to achieve for the group treated with drug X for both male and female subjects. In a study, it is possible to have more than one set of data being collected, which we labeled as data set 1 common. In our case, we only have one set, and it will include the collection of the outcome response. This information is used later in the data collection module. The timeline is where all the groups, treatment, factors, and responses come together to define the sequence of events within a study. The researcher creates an event description for each step in the study. A typical study will have a pre- and a post-treatment measurement sampling event, as well as a treatment event. The first event is the control sample event, which is associated with being performed on both the treated and untreated groups. Being the first event, we see that the time for starting the event is when ready. We next select the sampling data set to be used at this event. And finally, we define if the animal subjects are to be sacrificed after the event. Our next event to define is the treatment event. Here we only select the group which will be administered the drug X treatment. We now see a scheduled field in which we can set the time lapse between control sampling to the time of treatment. Here we set it for 1 day after control sampling event was completed. And finally, we say no sacrifice at this event. And then finally, we create a post-treatment sampling event that we do for both the treatment and non-treated group. Here, again, we define the time lapse desired between the treatment event and the post-treatment sampling event to be 3 days after treatment. And we select data set 1 common as the data to be collected, and finally, no sacrifice. With all the key design factors specified, the researcher can now move on to analyze the statistical options. Based on the structure of the specified study design, a decision engine recommends from a set of choices such as t-test, one-way ANOVA, factorial ANOVA, and several others, the best test type for the given structure. The intelligent analysis engine deals with your design complexities, such as nested designs with multiple factors and levels. An initial analysis is automatically run with common values for statistical parameters like significance, power, and effect size. And a graph shows you study power versus sample sizes for the study contrast. A table is also displayed to visualize the group interaction effect sizes, or their power, their sample sizes, and cost. At this point, the researcher may iteratively experiment with setting parameters to different potentially desirable values, and then observe the impact on the sample sizes and associated costs required to achieve these targets. The researcher may also experiment with having parameters other than sample size recalculated, such as power or effect size. We can easily recalculate a new sample size to meet a new target power for any of the study factor interactions. When the researcher is satisfied with the expected analytical results of the study, the review page presents a simple summary view of how animal subjects will be allocated across the data plan to achieve these results. When the design is saved and approved, you'll return to the home page and you can see that the data collection module icon is now enabled. You can also navigate to the Reports module to generate Word or PDF documents for use in proposals or submission to animal use approval committees. You can also share the design with others by going to the Team module. If a researcher has questions or areas of uncertainty and can benefit from speaking or working with somebody with specific expertise in statistics applied to preclinical studies, he or she can simply click the experts icon to access Seralogix service that accompanies the web application and connects the researchers with expert consultants who can provide guidance to improve study designs, improving the quality results and reproducibility. Live chat is also available when the researcher is in need of immediate help. Preclinical studies are a major undertaking and carry considerable risk of errors that impair reproducibility of study results, wasting time and money, and potentially even damaging reputations. Use of the Seralogix Study Manager significantly reduces the chance of these painful errors, and while simultaneously making studies smoother and easier to design and manage. Please proceed to view the data collection module demo to see how the design module seamlessly sets up data collection and facilitates the collection of higher quality data. For more information, please visit our website or send us an email at info@seralogix.com. Thanks for watching this video, and we look forward to hearing from you.